So DNA sequencing by standard method was gold standard for many years, and then we have had over 4 million donors using this technology. And as Paul alluded, standard sequencing has a limitation because it amplifies the sequence both haplotype from inherited from both parents. So the next generation sequencing gives you advantage of testing one single molecule at a time. And then all of them are based on the premise that you take one single molecule and then, but by various methods, you do the sequencing. There were, right now, there's 4 different technologies that has been tested in our lab and in the community, and I'll briefly explain to you. Illumina is the most popular method. It uses the glass surface here, and then a single molecule binds the surface, and then amplification of the single molecule happens on the surface, and signal DNA detection of the DNA sequence is done by sequencing by synthesis, and then each time one nucleotide repeats and incorporated ones are after the finish it gives a signal, and these are detected by camera. Basically, they take the picture and then putting after all these bases, it's get sequence information. This technology had a limitation at the beginning. It was not sequencing long regions, but now it's good enough that we can cover the exome by sequencing from both sides, 250 times 2 base pair. And other technologies based on emulsion PCR, the bead-based assay. So by emulsion PCR, they were hoping to get one single molecule on a bead and then amplify and then do detection. The 2 methods, 454 and then iron based on emulsion PCR. We stayed away from it because it was very labor intensive and was not suitable for high throughput. And another very popular method coming being heard is that by biosequencing, that this sequence, the advantage of this technology is that it gives you longer reads of sequencing, and it's based on single molecule real-time sequencing. And you get longer reads. So Illumina sequencing, what we do is that we target the exon here, gene-specific primers, you do one PCR adapters, and you do secondary PCR to add your adapters to go to the glass surface of the Illumina chip, which is called the flow cell. So basically, it's very straightforward and much easier to scale up than other NGS methodologies. So this is an animation that shows what's going on. Single molecule binds, PCR happens, and then cluster generated, and then this cluster is being synthesized, sequenced by different fluorescence nucleotide, and then you get the sequence, and then they get analyzed in the background. So all these things is done scaled with automation, and thanks to next generation sequencing, now we get high resolution typing result even for registry typing projects. And this column was SPT alone. We used many different applications to get to this level of high resolution, but as you see, almost 99% of high resolution achieved for multiple both-sided NGS. And we validated this by doing parallel sequencing, close to 500,000 donors for all both sides, and then right now we have 33 MySeq to do this work. So this became routine now in our lab. We do all other genes besides HLA, AQRA. Here also we routinely do. So we got the backfire sequence, and I will show you 2 animations produced by this. understandable, or you want to explain it?
Okay.
So there's ATCG.
Just turn it up.
Yeah, ATCG nucleotide there, and then they are. I will go faster.
This is the beauty of legends.
So basically, you capture one DNA molecule with one enzyme in a small environment, and you capture the signals here. So this is happening in real time, and you capture the signal in real time. This is like in parallel, and then you get very long sequence speed then in just in around 2 hours. So they claim you can do one genome under $100 and then under 1 hour, but this is not true yet. So we are using this technology to do full-length exon 2, 3, and 4 sequencing so that we can get all 3 exons in one phase. And this is important for phasing to deal with the recombinant alleles and then to get the most accurate results. So you cannot do this long PCR the way in other technologies, but only with plasmid bio. And we are using this in clinic to resolve certain ambiguity that's not being resolved with classical methods. So here we overlay exon 2 and 3 with the Illumina sequence plus PacBio, and then we get very good concordance. The beauty of this method is you can sequence in one shot 40 kb DNA sequences. This is a positive code of kill region. The straight line here is PacBio sequence. We sequenced the same clone with Illumina, and the Illumina sequence was a good concordance, but certain regions could not align. These are because of the highly repetitive regions and the kill region that other base pair regions they keep repeating several times, and no alignment algorithm can really deal with this. So the PacBio will give you good coverage. And then the last thing I want to show you is that this is the loss futuristic technology called next generation sequencing based on Oxford nanopore single molecule sequencing. So in nanopore is that these are nanopore are made by proteins, and then it's on a semiconductor chip. There is a membrane here. This indicates the cell membrane, so that they apply current. There's a single straight line, but when the DNA molecule passes, based on the base, the ACGT, there's current interruption, and these are detected as a sequence. And the very small instrument, you can look at the outcome.
Potential is applied across the membrane. flowing only through the aperture of the nanopore. Single molecules that enter the nanopore cause characteristic disruptions in the current. By measuring that disruption, the molecule can be identified. In strand sequencing, an intact DNA polymer is sequenced as it passes through the nanopore. Here you see a DNA enzyme complex approaching the nanopore shown in blue. The enzyme shown in green is designed to ratchet the DNA strand through the nanopore one base at a time. The enzyme binds to the end of the double-stranded DNA and unzips the double strand to form a long single strand which it feeds through the nanopore. As the DNA strand moves through the nanopore one base at a time, a characteristic disruption in current is created by the presence of particular combinations of bases in a particular part of the nanopore. Because these disruptions in current are so specific to the different combinations, this information can be used to determine the order of bases on that DNA strand. There is no deterioration of accuracy as the long DNA strand is sequenced. By preparing the DNA so it has a hairpin structure at its end, the system can read both strands, that is the sense and antisense strands of the DNA. This gives advantages in data analysis. In order to create a high-throughput system, a number of nanopore experiments can be conducted at the same time by using an array chip. Multiple microwells are fabricated onto an array chip using standard semiconductor materials. These array chips may be scaled according to need. The user may require tens of channels to hundreds of thousands of channels depending on the application. This array chip is built into a consumable cartridge, which also contains the fluidics required to run the chip. The analyte is added to the cartridge, which is then used in conjunction with an instrument, the gridiron node, for real-time data collection and analysis. The array chip is also designed to be used inside a miniaturized RNA instrument.
So we did not experiment. Several experiments using nanopore. We used the phosphate clone I showed you and did it with backbone. You get the result under real time when the signals goes to the cloud, and you get the reading as the reaction goes on. The principle is really very neat. However, we have a problem with accuracy here. This technology is still not perfect. This is a phosphate clone that we had sequenced and nanoparse very perfectly overlaid, but when we zoom in, we see all of the gaps and that mismatches. So the match rate is around 60 to 70% based on the phage or phosphate clone we use, and then gaps around 10 to 15%. So this is still not acceptable for clinical use. And again, the long HLA product with sequence with backbone, when we compare, we see similar things around 60 to 70% identity with the real sequence. So it's a promising technology, but it still has a lot to go. So that's all my update about the next generation sequencing technology. Thanks for the attention.